chromosomes in this cell? Is this a diploid or a haploid cell?
No, it's
Diploid. I have two sets. I have a pink set for my mom, and I have a set for my dad. Diploid, two sets of chromosomes. So my 2n for two sets equals how many? 4. 2n equals 4 for this cell. For humans, 2n would equal 46. And n would equal 23. So here I have, where would this cell be in the cell cycle?
G1.
G1. So now, if it needs to divide to make gametes or sex cells, I'm going to duplicate some chromosomes. So they duplicate during which part of the cell cycle? Now here's some duplicated chromosomes. So 2n equals 4. Still 4 chromosomes. Now, what can you say for the amount of DNA? The amount of DNA doubled, right? So we still have 2 equals 4. Now, if I'm doing meiosis, now in mitosis, what would happen is these would all move to the middle of the cell, right? That'd be mitosis, and they'd separate, and we'd end up with 2 identical cells that all have 2n equals 4, right? But that's not what happens in meiosis. So, what is the first step that happens, class?
they line up again.
What are they going to do?
They pair with their homologous pairs.
They find their homologous pairs. So now I have some traits on some chromosomes. This is a trait for eye color and dimples. This says green eyes and dimples. It says blue eyes and no dimples. They're both the same traits, even though they're different versions. So up here, this would find its homologous pair. And then here, I have hair color, red or blonde hair, short, height, or tall. Height and hair color, that's another homologous pair. Now guys, when they pair up right here, could it very easily end up with this one on this side?
Yes.
Totally possible, right? Isn't it totally possible?
Yeah.
Okay. Now, here I have a tetrad. Now, in real life, this sister chromatid is like a zipper, zipped to this sister chromatid. They get really attached. Really attached. And then what happens is they're going to exchange parts of their DNA. So what happens is they're zippered together, so they're just going to like shuffle. They're going to shuffle traits. So here, the tall gene for height gets switched with the short gene. So now it's still a tetrad, but we've exchanged sections of our chromosome with each other. Now, did I change a tall with the red hair? Did I change different genes?
No.
I exchanged the exact same trait, the exact same gene. So is that okay? Could I add anything extra to my chromosome? No. There's just a little bit of shuffling going on. So now here is still a tetrad, still homologous pairs. But what can you say about my 4 sister chromatids? They're all different. Aren't they all different now? They're all different. Before I had 2 identical and 2 identical. Now I have 4 different. So did crossing over contribute to variation? Yeah. It created I've seen different statistics. In your book, I think it said it happens 1 to 3 times on each chromosome pair, or like this. But I've also seen where it happens like up to 50 times. Because there's 1,000 genes. You can really like separate a lot of times. Anyway, and up here, crossing over would happen. But crossing over pretty much always happens. Now, this is what wasn't in my video, because that would have been just too hard to
Make.
Yeah, and then to keep them all moving. Nope, sorry. So anyway, what's my next step? Wait! With your homologous pair, please do crossing over. Yeah. Are you recording it?
Yeah.
Did you hit stopper now?
Oh, no.